here's a time lapse of a frog embryo putting its face together. And what you will see, and this is one frame out of that video, what you will see is that this is something we call the electric face, discovered by my colleague Danny Adams. When she was in my group doing this profiling, she found that here's a particular frame from that video where you can see the animal's right eye, the mouth, the placodes. All of this is already demarcated before the genes start to come on and really pattern the face. This is a bioelectric pre-pattern of what the future face is going to look like. And I'm showing you this one because it's so simple. It literally looks like a face, right? There are other patterns that are much more complex that you can't really just, sort of visually decode. But this one is very clear. This is the memory of what a tadpole face is going to look like. And if you move this, if you disrupt this electrical pattern, the gene expression will change and the organogenesis will change. I'll show you examples of that momentarily. So this is an endogenous pattern that is required for normal development. In fact, human channelopathies human patients that have craniofacial defects often have mutations in ion channels that screw up this pattern. Now that's a normal pattern. Here's a pathological pattern. We've put in a human oncogene. These cells will make a tumor, but even before the tumor becomes apparent, you can already see this aberrant bioelectrical signature of these cells basically disconnecting from the rest of the tissue and going to their unicellular ancient behavior mode, where the rest of the body is just environment as far as they're concerned. So one set of tools is to track these bioelectrical changes. Now here's another important set of tools to actually start to be able to write new information into it. And the way we do that, we don't use any applied electric fields. There are no waves, there are no magnets, there's no electromagnetic radiation. What we do is we do exactly what neuroscientists do, which is we modify the endogenous mechanisms by which cells establish electrical signals. So either ion channels, we can open them, we can close them, we can use light or drugs to do that. And the gap junctions, the electrical synapses. So we can determine which cells talk to which other cells by opening and closing these little gap junctions. It's very telling that all of the tools of neuroscience work in other cell types. Basically, the tools can't tell the difference, right? This distinction between neuroscience and other cells in your body is completely artificial, and it's a consequence of humans trying to parse the world. It's not. Nature doesn't obey that distinction. So what can you do with this? Okay, I've been talking about this electrical information. What does it actually do? Well, here are a few examples. One thing you can do is you can take this tadpole, and I've shown you the electric face. And so we ask the question, okay, if now that we know that there's a particular voltage state that kickstarts eye development, could we simply reproduce that somewhere else? So we took an ion channel RNA that we knew was going to set the same voltage state. We injected it into precursor cells that are going to give rise to the gut. And sure enough, those cells can form a complete eye. The eyes that are formed will have all the right layers, retina, nerve, optic nerve, all that, lens, and they can be formed anywhere. They can be formed anywhere in the body, as long as the cells get the right pattern that tells them what organ to make. Now, notice two very interesting things here. One is that this instruction is highly modular. We provided a very small piece of information, just a voltage pattern. We certainly did not give it all the information needed to specify an eye. Eyes have many cell types. They're very complex. We have no idea how to micromanage the creation of an eye. But what we provided was a kind of a subroutine call, a signal that says build an eye here, and the cells do. That's the first thing. The second amazing thing is that there are two levels of instruction here. One is us instructing the cells build an eye. But here, what you can see is this is a lens sitting out in the flank of a tadpole somewhere. And the blue cells are the ones that carry this ion channel that we put in. But there's not enough of them to make a good lens. So what they've done is recruit their neighbors. This is why at the very beginning I pitched this idea that frogolotls might have legs that consist also of frog cells. Why would frog cells build a leg when they normally don't? It's because cells have the ability to instruct each other about what to build. And so these cells right here, they've been told to make an eye, but there's not enough of them, and they know that. And so they recruit a bunch of their neighbors, these brown cells that we never touched. They're not, these cells are completely wild type, and yet they're participating in making this ectopic lens out in the tail of a tadpole. So there's this amazing ability to instruct, and for the cells to then instruct each other. Now, what else can we make? Well, we can make, using the same methodology by manipulating these ion channels, we can make otocysts, which are balance organs, like inner ear type of organs. We can make complete beating hearts, so ectopic hearts. We can make extra forebrain, and then you can ask if these animals are any smarter than the normal tadpoles. We can make ectopic limbs, so you can see here all kinds of extra legs that we can form, and we can even make fins. Now this last one's kind of weird because tadpoles, of course, aren't supposed to have fins. We'll get to that momentarily. But what you can see is that by changing the bioelectrical pattern using manipulation of ion channels, you can tell this collective where to go in morphospace, meaning what types of organs they should be building. You're not micromanaging the specifics, each step that they take. They figure that out on their own. In fact, we have no idea how to do that for any of these complex organs. But you can indicate regions about where it is that they should be going. And this has biomedical implications. So frogs, I told you, unlike salamanders, do not regenerate their legs. So if a leg is amputated 45 days later, there's nothing. What we've done is come up with a drug cocktail that targets ion channels that induces leg regeneration. And so you can see here, 45 days later, there's already a nice leg forming. The leg is touch sensitive. It's motile. It's functional. It has a pretty good final pattern. And so we're in the process of translating that to mammals. Hopefully this will work. I have to do a disclosure here because this is a commercial venture now. Morphoceuticals Inc. is a spinoff between myself and Dave Kaplan, where David's lab makes these bioreactors that that have an aqueous environment for the wound, and then we provide the ion channel and other types of payload in there to hopefully get leg regeneration in mammals. So this is a clinically clinically relevant approach. So I want to switch from this now to show you another kind of amazing example of what bioelectricity does. These are Planarian. So I've already told you they have incredible amounts of regenerative ability. Because of that, they're actually immortal. There's no such thing as an old Planarian. They do not age. So these animals pretty much go on forever. And that's because of this incredible ability to continuously replenish and regenerate to keep their pattern perfect. So I want to show you an example. This is a Planarian, one head, one tail. You cut off the head and the tail, you got this middle fragment. We check the gene expression. Yep, anterior genes are in the head, no anterior genes in the tail. That's fine. And 100% of the time, it makes this normal offspring. So one head, one tail. Okay, it regenerates. Now here, what I'm going to show you is that here's a Planarian, one head, one tail. Again, anterior genes where they're supposed to be. But when I cut this guy, he makes a two-headed form. Now, I just told you that this process was very reliable. Why in the world would a one-headed animal make a two-headed? And this isn't Photoshop. These are real live animals. So here are heads at both ends. Well, it's because in the meantime, we found that there's an electrical circuit that actually helps this tissue remember how many heads there's supposed to be. And what you can do in the meantime is take this one-headed animal and you can look at the electrical pattern. The electrical pattern says one head, one tail, and we can alter that electrical pattern to say, no, you're going to be a proper worm should have two heads. Now you can see it's kind of a mess. The technology is still being worked out, but it's very clear. We can impose this pattern that says two heads, and when you cut that animal, boom, that's what the cells make. Now this is really critical to understand. This bioelectric pattern is not a map of this two-headed guy. This bioelectrical pattern is a map of this one-headed animal into which we incepted a false memory of what planaria are supposed to look like. And they sit there, this memory sits there latent, doing nothing until you injure them. Once you injure this animal, that is when the cells will pay attention to this pattern and they will go ahead and make a two-headed animal. So if you're wondering where counterfactual, where the ability to consider counterfactuals came from, this kind of mental time travel where you can consider things that have memories and have predictions about things that are not happening right now. This is perhaps the sort of basal precursor of that ability in morphospace. This animal has a representation in morphospace of where it will go if it gets injured at a future time. Not what's happening right now. This is not where I am right now. This is where I'm going to go if I get injured. Now, why do I keep calling this thing a memory? I keep calling it a memory because if you take these two-headed animals and simply recut them again in plain water, no more manipulations of any kind, they will continue to rebuild two-headed animals and forever, as far as we can tell. The question of how many heads a Planarian has is not strictly determined by the genome, because we didn't touch the genome. The genome is wild type. There's nothing, there's no genomic editing here. That information is sitting in an electric circuit. The default, the genetically set default, is one head, but it's easily changed. And in fact, we can now, well, now it's, I say it's easy. It took us 10 years to figure it out, but in fact, it's quite readily done. And in fact, you can set it back. You can set it back to being one-headed by again targeting the information in that electrical circuit. The first time I showed these data, somebody stood up at a conference and said, "Well, that's impossible. Those animals can't exist." So I always bring this video so everybody can see what the second and third generation of these guys look like. So this has all the properties of memory. It's long-term stable. It's rewritable. It's got a little bit of lability, so it's rewritable. It's got latency or conditional recall. So you can have a single body can have memories of what a planarian is supposed to look like that aren't true right now. And it has discrete possible outcomes. So one of the things that we're doing now is really trying to integrate models of the physiological state space of this animal, that where the circuit, the electrical circuit has a state space where there are attractors that correspond to one heads, two heads, no heads, and so on, and merge that with models of connectionist artificial neural network type of processes where we already know how to build networks that, for example, do pattern completion, and they have a memory of what the pattern should be, and if part of that pattern is missing, they can reproduce it, and so on. This is all part of one problem, and the key is tying it all together with these quantitative models that show how this electrical circuit navigates morphospace. Now, what I've shown you is that you can tweak that electrical circuit to find an attractor that has two heads instead of one head. But what else is there? What else does this space actually have? Let's explore that a little bit. Well, the first thing you find is that, well, it contains head shapes of other species. So what you can do is you can take this planarian, cut off the head, perturb the electrical circuit for about 48 hours, then let it go. And when it finally settles down into the correct attractor, it doesn't always find the right one. So some percent of the time it goes back to normal and makes this nice triangular head from D. doratocephala, but sometimes it'll make a round head like this S. mediterranea. Sometimes it'll make a flat head like this P. felina. In fact, this is a stochastic process. The frequency of these is proportional to the evolutionary distance between the real species that are being mimicked here. And again, no changes to the genome. There's nothing genetically wrong with these animals, but you can find these other attractors, and it's not just for head shape, it's the shape of the brain. The distribution of the stem cells become just like these other species. And they're about 100 to 150 million years distant. Okay, so you can find those attractors with the exact same genetics. In fact, you can find shapes that don't correspond to any real planarian as far as we know. You can make these crazy spiky forms. You can make a different symmetry type. So this kind of cylindrical radial symmetry instead of a flat bilateral symmetry. You can make combinations where there's a flatworm with a big tube growing out into the third dimension. And again, nothing wrong with the hardware. There's no mutations. There's nothing wrong with the genome. It's bioelectrically exploring the space of where it can be. And so the idea is what we're doing now is building these kind of full stack models that try to integrate all the way from the molecular information about what channels do you have to work with, so the hardware, all the way up through the physiology of, okay, well, what does this mean for the voltage states across the tissue, to, well, what does that mean for the identity of different organs along the primary axis, to what kind of algorithm is this whole thing implementing such that we could actually understand the algorithm and intervene, right? So make changes. And for this, we have all kinds of computational platforms such as this, which allows you to load up electrical circuits into cells and then simulate tissue and organ level dynamics to understand what the large scale computations are. Okay. And so just as a simple example, again, of why this is useful and what the practical implications of this are, we use this strategy to find a technique for repairing birth defects. So here's a tadpole, here's the forebrain, midbrain, and hindbrain. If it's exposed to nicotine or various other teratogens, you can see there's massive defects. So the brain is basically gone. The forebrain is basically gone. The midbrain and hindbrain are damaged. And so what we did is we built this bioelectrical model that tries to explain how does the brain know what its size and shape should be. And in cases where, for example, here, this is even worse than a teratogen, we mutated a key neurogenesis gene. This is a gene called Notch. So this gene is mutated. You can see the forebrain is just gone. The midbrain and hindbrain are a bubble. These animals have no behavior. They just lay there doing nothing. It's a very strong defect. What we did was we asked that model, which ion channels would we have to open and close to get back to the correct bioelectrical memory of what a correct brain should be? Basically, how do I find my way back in morphospace to what a correct brain should be? And the model made a prediction. We tested that prediction. It happens to be an already human-approved set of drugs that can do this. And sure enough, when we did it, there you go. Their brain structure comes back. Their brain gene expression comes back, and their IQ comes back. So if you actually test them for their learning rates, they're indistinguishable from controls, even though they've been exposed to teratogens or they have this really nasty genetic defect. Some problems, and not all problems, but some problems can be fixed at the level of software. Sometimes when your hardware isn't quite right, you can make up for it in software by having a better navigation policy through morphospace. So you're not automatically dead in the water if your hardware is a little wonky. So what that means is we ought to be able to create these pipelines and we've already started to help design electroceuticals. That is, you have some particular problem, and you can ask the simulation, what type of channel, meaning what drug, can I use to get back to my correct bioelectrical pattern? Fundamentally, this is really the question of how do we exploit this electrical interface that these cells are exposing to us, right? Just like neurons do, and people using electrodes and optogenetics and everything else to control brains using this electrical interface. We can do exactly the same thing to help guide their navigation through morphospace. It's a new way to think about making drugs for various disorders of development, of injury and cancer and so on.